Well, no, you definitely do, Nelly.
Yeah, I know. The type A, oldest neurotic child. That should be one of our future papers. Somebody could do a paper on birth order and what it all means.
See, Google Scholar here.
Hey, Marion, how you doing? You're at home. No chemistry class today?
No, it just ended early, so we got to leave. That's like the best thing.
Yeah, they build in the extra time.
Yeah. Yeah, we needed our product to dry, so it was like can't really do our
Oh.
other tests yet, so
Under vacuum oven, just room temp.
yeah, we did like vacuum filtration and then he wants us to get the like infrared spectrometry and melting point and that kind of stuff. So we have to let it dry first.
Don't miss that.
Uh, how are you, Allison?
Good. I had a nice spring break. It was nice to have some time to relax.
Did you go somewhere?
I stayed local, but we had like a little staycation. We stayed at a hotel to feel like we had a little bit of a change of scenery to feel like we're somewhere far away, but no, we are still close to home.
Oh, it's still nice to get away. It was nice to have the week off, I'm sure. Yeah.
Mhm.
Pancho, you doing good?
Yeah, I had a pretty relaxing break. I did get sick halfway through it, and I'm still kind of sick, so that kind of sucked. But I mean, definitely took the time to relax. I picked up. I learned more how to do stuff with video editing over the break, so
Oh,
pretty good.
What program do you use?
I'm trying to learn how to use this program called DaVinci Resolve. Um, and so far it's pretty complicated, but it's free out the box and has a lot of features, so definitely worth it.
Do you make a bunch of videos? I didn't know this.
It's mainly just for fun. Pretty much just yeah. I am just mainly just trying to learn the basics and then go from there. But yeah.
That's awesome.
I tried to teach myself Adobe Premiere Pro. It wasn't fun.
Yeah, I just use iMovie if I like create a little video of like, I don't know, a fun adventure.
Yeah, the learning curve for like self-teaching editing is like really high. It takes a lot of time before you can even get some basics done. But I think I'm at the other end or getting to the end where I can actually, produce some decent content. So yeah.
I use some free program I don't even know the name of that probably put a virus on my computer. um, but it didn't take long to learn it, but maybe the security features on my computer are compromised as a result. But what can you do? Um, Aneth, I don't know if you want to introduce Andy or should I or okay.
Yeah, 10 years this May. Can't wait. Fair enough. Thank you, Ananth. Um, so yeah, Ananth, I mean, I knew about the club, and Ananth had approached me, and I'm thinking nobody wants to read any of my articles. I mean, a lot of it's just gonna be. I'm not trying to be here over people's heads. So like, I just brought up my Google Scholar library here. So I think here we go. Engineering a synthetic anaerobic respiration for reduction of xylose to xylitol using NADH output of glucose catabolism by E. coli. That's the sort of crap I used to publish. So instead, I thought, hey, why don't I give my old dissertation defense presentation with some tweaks? But yeah, so I'll try to be as quick as I can. I don't. One of the things I dreaded about seeing speakers talk about their work was they would agonize over like minutia, and it was awful. Nellie's nodding; she knows exactly what I'm talking about because it's this one diagram is super important to them. They're like, I gotta talk about this for half an hour, and no one cares. So I'll try to be concise here. But yeah, this is the presentation I gave, which kind of gave the culmination of what I did over about 5.5 years. PhD projects, they don't have an end until your committee says it has an end. Like I tell a lot of people get master's degrees because it's 2 years, you take some classes, you do a small project, you write a thesis. No one's going to keep you there forever. With PhD projects, you are done when they say you're done. And for me, it was 5.5 years, but I've known people took a decade. It's quite the process. But anyways, my background is primarily microbiology. Took lots of microbiology classes, a lot of molecular biology classes, and I got teamed up with who was a new professor at the time, and he wanted to work on biofuels. And I'll get into this more in a second, but we worked primarily with E. coli, and we were what's called metabolic engineers, where we would genetically manipulate E. coli's DNA so we can control what sort of useful products it could be capable of producing. So the idea is you feed the organism sugar and it craps out money. Okay, so that's basically what we did. Oversimplified. But anywho, and stop me if you got questions. Like I said, I'm going to be kind of quick about things. Because I barely care about this stuff myself. I'm kidding. Mostly. And let me tell you, it was real weird looking at a PowerPoint I haven't touched in a decade. It's like, ooh, I've gotten a lot better at this, so it's okay. So. But anywho, so this is kind of what the time of my project was over 5.5 years, and I'll explain what it all means. But we dabbled in increasing what's called reducing power output in E. coli to increase its product yields. I'll talk about that bit here momentarily. I'm also going to make one assumption, and I should talk to Nellie about this before. I'm assuming we've all had at least a basic biology class at some point. I know Anant has. You have no excuses, sir. But the rest of you, I don't know yet. But anywho, I know someone's got chemistry, so that's good. All right, so like I said, my work was in metabolic engineering, and when I talk about metabolism, it usually makes a lot of students shudder because, if I ask students, what was your least favorite chapter in biology? It's usually the metabolism chapter. So respiration, maybe photosynthesis, depending on if you had Nellie or not. But anyways, so respiration, or at least aerobic respiration, is this process where cells will take sugars like glucose and slowly strip it of its energy. Okay, we oxidize organic matter. And as we strip our organic matter, we take that energy, those electrons, and we're ultimately using them to make ATP. Okay, so in Gen Bio, they tell you, right, 1 glucose has enough energy in it to give you either 36 or 38 ATP, depending if it's prokaryotes or eukaryotes. And this process requires oxygen, at least for aerobic respiration. Okay, so as we strip, say, glucose or sugars of their energy, that energy is getting transferred to a molecule called NAD. Okay, you hopefully have heard of NAD in the past. It's a type of coenzyme. It's this molecule whose one job is to be an electron taxi. Okay, so as we strip glucose of its energy, we're transferring that energy, whoops, come on, stylus, to make these NADHs. Okay, so in total, one glucose has enough energy to be transferred to make 10 NADHs. Okay, and those 10 NADHs deliver their electrons to the electron transport chain, and that's where you make the bulk of the ATP. Okay, but hang on to this number, 10 NADH. Okay, so that's the metabolism chapter in a nutshell. All right. Oh yes, and these reactions where we harvest energy, transferring to NADH, we call them redox reactions. All right, now, I didn't really do much work with anaerobic or with aerobic respiration. My work was primarily with fermentation, a topic near and dear to me. Most people have heard of the term ferment or fermentation before, and fermentation is how organisms live without a means of respiration. Okay, so looking at E. coli, for instance, E. coli can use oxygen for aerobic respiration, but if you take away oxygen, it doesn't suffocate and die. It switches to fermentation. So I gotta move my little screen here. All right, so the idea behind fermentation is to regenerate the NADH supply. Okay, because without oxygen, without an electron transport chain, there's nowhere to deliver the electrons. And eventually you get an NADH-NAD imbalance. Okay, you have too much of the reduced form, NADH, and like none of the oxidized form, NAD. And then the organism dies. With fermentation, it's a means of keeping that NADH-NAD balance going. Okay, so remember your metabolism here. Glucose goes through a process called glycolysis, where it's turned into 2 pyruvates. Now, normally, those pyruvates would then go into the Krebs cycle as acetyl-CoA, okay, or the TCA cycle. With fermentation, instead, what happens is that these pyruvates go through a different redox reaction, becoming tasty, tasty fermentation products. Okay, and these fermentation pathways help to regenerate that NADH-NAD balance, so the organism can keep going. It comes at a cost. You don't make nearly as much ATP with fermentation. You only get 2 instead of 38, but it's still a means of living. They're basically living in poverty, but they get by. Okay, so again, there's all this stress on NADH, right? So in a cell, the NADH and NAD levels have to be balanced, okay? With aerobic respiration, with electron transport and oxygen, right? NADHs drop off their electrons at the electron transport chain, and they get dumped on oxygen at the end, okay? With fermentation, okay, we're relying on these fermentation pathways instead to reoxidize NADH to NAD and keep this balance. Now, here's where fermentation gets really neat, okay? Lots and lots of bacteria love to ferment, okay? Not all of them do, okay, but many do. And the neat thing is that all fermentation pathways begin with pyruvate. And then, depending on the organism, they can branch out to all sorts of different products, many of which we consume, okay? Yogurt is, and you can see this on the side of the yogurt container, it'll say contains these active cultures, right? Lactobacillus, Pediococcus, trying to think of others off the top of my head, mostly lactobacillus, but we starve them of oxygen, they crap out lactic acid. It's what makes yogurt sour, okay? Swiss cheese is inoculated with Propionibacterium. That neat tang you get is a mixture of propionic acid and vinegar. My micro teacher, Dr. Rangaswamy Meganathan, always used to love to tell the story about tequila. Okay, most spirits are made with yeast, but tequila is unique in that it's actually produced through a bacterial species called Zymomonas mobilis. It's the only hard liquor that's actually produced through bacterial fermentation. So, fun fact. But you got all these bacteria with different fermentation pathways, all producing different products, just to accomplish the same thing, to keep surviving in the absence of oxygen, to keep regenerating that NADH-NAD balance. All right, so why do we care? Okay, that's the important part. So it turns out many fermentation products are of commercial value. Okay, ethanol, right? So corn ethanol, that's a biofuel. That can be the product of large-scale fermentation. One that another person in my lab dabbled with, which was an awful experience for them, was butanol. Butanol was actually looked at as an alternative to gasoline back in the 1950s because it has the same potential energy as gas. It's just a real bear to produce, but there's a group of bacteria that will produce it naturally. They'll ferment sugar, crapping out something has the same energy as gasoline. But the list goes on. Lactic acid. Okay, a lot of biodegradable plastics are made of PLA, polylactic acid. Again, that's something bacteria produce naturally. But in my lab, we dealt with looking at two major fermentation products. We were looking at the production of ethanol, and later on, I looked at producing xylitol. Okay, so engineering bacteria to ferment and produce rare sugar alcohols. Let me check one side. Okay. So here's the overall idea. Okay. You start with some microbial biocatalyst. Okay. You take some easy to work with organism, and then you genetically modify it to produce some fermentation product. Okay, so if you have a difficult to work with organism that makes butanol, like Clostridium acetobutylicum, you just take the genes from that organism and you shove them into E. coli and say, hey, E. coli, you start making butanol now because you're cheaper and easier to work with. There's been a grand debate for a long time, kind of like the holy trinity of microbial biocatalysts have been bacteria, yeast, or algae. And last I checked, I think algae is still winning because you can just really bulk them up with biofuels. But anyways, you take your microbial biocatalyst, bacteria, yeast, whatever, you feed them basically plant material like corn. I won't get into details here of how that gets broken down, but ultimately when you break down plant material, you end up with a mixture of sugars. The genetically modified organisms take the sugars and then they crap out money. All right, so we focused on E. coli as a biocatalyst, okay, for a bunch of reasons. For one, E. coli isn't a particularly picky eater. It will use a variety of 5 and 6 carbon sugars, which is great because biomass is made up of lots of 5 and 6 carbon sugars. It grows very cheaply. Grows in minimal salts media, which is very inexpensive. It does have the ability to produce fermentation products at high yields. Let's see what else. It's fairly tolerant to substrates and fermentation products. That's something I won't have time to get into, but that's something another student in my lab focused on, which I can talk about later if you want. But most importantly, it is a joke to genetically engineer. It's so very easy to genetically manipulate E. coli. But those are the big reasons why we chose to work with E. coli as a biocatalyst. How am I doing here? All right. I feel like I'm just plowing through. Meh. We'll skip that. So this is just more talking about like the different approaches to this. So when you want to design a microbial biocatalyst and genetically engineer it, there's two major approaches. One, the traditional one, is to kind of add genes or delete genes and just kind of force the flow of carbon. The other way, which we focused on, is cofactor manipulation, increasing the amount of NADH so you force it to make more fermentation product. Okay, so one of the hurdles with E. coli we had to deal with is that natural, wildly occurring E. coli actually makes 5 different fermentation products. I always remembered it as false. Formic acid, succinic acid, acetic acid, lactic acid, ethanol. I didn't do those in the right order, but still, it's false, okay? And these products are kind of all over the place in their NADH, NAD demands. Basically, it all balances out. So ultimately, what we'll do here is that we want to eliminate these other fermentation pathways. We want to only produce ethanol. The problem is that ethanol costs too much NADH without the other fermentation pathways in place. So you end up with a deficit and the organism could die. So we had to figure out how to get around that hurdle. How do we get it more NADH so that it will make ethanol for us and not just crap out? All right, so as I just mentioned, some fermentation pathways require more NADH than others. Okay, so lactic acid, for instance, lactic acid requires 2 NADH. Ethanol requires 4. So problem is that with glycolysis, you only get 2 NADH. Okay, and you need 4 to make each ethanol. So again, there's a bit of a deficit there. So our goal is to somehow genetically manipulate E. coli so that we increase the NADH output, say get 2 more, so we can balance things out and make ethanol. All right, so long story short, we increased NADH output by doing 2 major things. So first off was the anaerobic expression of the pyruvate dehydrogenase complex, which I'm sure none of you have heard of, but you may have heard of the transition step. So you go through glycolysis, you get your 2 pyruvates, and then they talk about this transition step into the Krebs cycle, where it becomes acetyl-CoA. That transition step between glycolysis and Krebs, that's the PDH complex. Okay, so we're going to get 2 more NADH out of that. And then if I have time, I keep looking at the clock there, we're going to squeeze out 6 more NADH by genetically modifying the TCA cycle. The problem with both of these steps, both of these pathways, is that they don't work when there's no oxygen. Okay, and if we want to ferment something, we need to take oxygen away. So we need to figure out how are we going to get these things to work under anaerobic conditions. All right, pictures are great, right? I got lots of hand waving here in text, but whatever. So this is what normally happens, right? If oxygen's around, this is what happens. You start with glucose, it goes through glycolysis, you get 2 NADH. The pyruvates go through the transition step, that's the PDH complex here, that's 2 more NADH. The 2 acetyl-CoAs go through that Krebs cycle, that's 6 more NADH for a grand total of 10. This is if oxygen is present. Problem is, if oxygen is absent, everything stops here. Okay. So, we did that one. All right, so ultimately our goal, like I said, is to produce ethanol as a sole fermentation product from E. coli, right? We want to feed it sugar so it craps out just ethanol and not the other letters and salts. So first thing we did, first half of the project, was to engineer homoethanol fermentation pathway. Okay, oh, you know what? I'll just get to all this here in a second. I think I've got like doubles of these. All right, so I look at my project as 2 halves. The first half was engineering E. coli to produce ethanol as a sole fermentation product. The other half I won't even mention unless there's time, okay? But again, for this project, to develop a homoethanol pathway in E. coli, we needed to double the amount of NADH in the absence of oxygen. Okay, so trying to remember here. I went over this, but I haven't done this in years. Go back. Here we go. All right, so some challenges here. Okay, so for one, of the 5 fermentation products, E. coli does not naturally make very much ethanol. Okay, because it requires the 2 extra NADHs, and those are going to be used by other fermentation pathways like making lactic acid. E. coli really likes to make lactic acid. So to meet this challenge, to take E. coli and say, hey, you're only going to make ethanol, and we're going to give you the extra NADHs, what we had to consider here was, one, we needed to eliminate the competing fermentation pathways, right? So that only ethanol was left. And then 2, we had to double the amount of NADH. All right. So here's kind of the overall goal again. Start with glucose, that becomes pyruvate, and then in a couple of steps, the pyruvates are fermented to ethanol. But again, this costs 4 NADH. Now, I'm getting ahead of myself here. So the first thing we had to do was to eliminate the competing fermentation pathways. Okay, so goodbye lactic acid, goodbye succinic acid, goodbye formic acid, goodbye vinegar, acetic acid. And you may wonder how we did that. We did that by doing gene knockouts, gene deletions. So I may just skip over it here because it'll probably take too much time. But we used something I don't know if folks use anymore. It's called the red recombinase system, where we used a kind of a targeting system to target each of the genes that make the enzymes that produce these fermentation products, and we physically remove them from the genome. And fun fact, if you completely remove a gene without any extra DNA left behind, the government does not require you to label it as transgenic or recombinant. Nice little loophole there, so I didn't have to file a bunch of extra paperwork. But anyways, so yeah, one by one, we knocked out each of the genes responsible for making the competing fermentation products, so that we were left with just ethanol as a fermentation product. So this is our old red recombinase gene deletion system, developed in the, let's say, 1990s by Datsenko and Wanner. I think they were in Indiana. Can't remember. Yeah, it's ringing in the back of my mind. But anyways, I'll get into the data here. But anyways, it utilizes 3 different plasmids. The problem is I don't know if folks know what plasmids are. They're these little, basically mini chromosomes that are primarily found in prokaryotes, found in eukaryotes, but mostly in bacteria. But anyways, these were specialized plasmids that allowed us to target specific genes and remove them, delete them from a chromosome. So let's get to the good stuff here. Talking about my data, right? Does this actually work? All right, so in the left column, those are our different strains. Okay, E. coli B was our wild type, our control. Okay, unmodified E. coli. And then each of the other strains, you can see the genotype here. That illustrates which of the fermentation pathway genes have been removed. So as it keeps adding them on, we're accumulating those gene deletions. So some of the neat things here. So we look at the final one here, SZ420. That's our homoethanol strain. Under anaerobic conditions, it actually grew better than the wild type. That 2.16, that's a measurement of how cloudy its culture became over time. If you ever have to do growth curves, I feel bad for you. Basically, you inoculate some broth culture, and you measure how cloudy the tube becomes over time. I got a picture of one later on, but I will tell you, we used to have to do these growth curves in teams because it would take like 36 hours. So someone had to take an overnight shift where every hour they would take these little tubes out, and they put in a little spectrophotometer machine and record the number. And we'd have like 72 of these stupid things at once. It was annoying. Anyways, so yeah, our homoethanol mutant grew best. Its final pH was the highest because it wasn't making competing fermentation products anymore. Ethanol is not an acid. And the neat thing is, everything's neat here, right? You can see the rest of the columns show you as we deleted each gene, it did stop making that product. They weren't measurable anymore. And in the final column, the ethanol output, it was 44 millimolar solution at the end, compared to 7 with the wild type. So what we were doing was working. Deleting the competing fermentation pathways was increasing the ethanol output. All right, now something I'm leaving out. How did we ultimately, I write too much on this stuff. How did we squeeze out these extra 2 NADHs? Because you need 4 to make ethanol, but glycolysis only gives you 2. That's where we got really clever, and we made that transition step anaerobic. Okay, so we took this incredibly complicated operon, and we switched it from being strictly aerobic to being strictly anaerobic. So that one step, that transition step between glycolysis and Krebs, is dictated by 3 separate genes that are kind of clustered together. So we call it operon. And all 3 of these genes are controlled together by a promoter. Now, normally, if you learn about promoters, you're learning about transcription, and they tell you, okay, here's the promoter. It's right next to the gene. That's what RNA polymerase looks for. But this is a dirty secret they don't tell you. Not all genes or operons have one promoter. The one we're going to talk about actually had 7. So I realized I just got ahead of myself because I haven't given this talk in a decade. But anyways, so this operon is regulated by an aerobic promoter. So it's only active when oxygen's around. Okay, oxygen's absent, no more transition step. So we said, okay, what if we switched out the promoter? What if we looked somewhere else for an anaerobic promoter and we switched them out? Maybe that would turn this operon on under anaerobic conditions. To this day, I still don't know why my advisor chose these promoters to look at, but he did. So the alternative pathway to the transition step under anaerobic conditions is called the pyruvate formate lyase pathway. Okay, this is a very, very anaerobic operon. So these genes only work under anaerobic conditions, and the super weird thing is they have 7 promoters. So we had to figure out, okay, which of these 7 promoters are actually worth taking and replacing with our pyruvate dehydrogenase complex? So using that same red recombinase system I mentioned earlier with the plasmids, we actually investigated a bunch of the different promoters, looked at different combinations, and we kept switching them switching out the promoter in front of that pyruvate dehydrogenase complex. And this is something we got a paper out of. This was a lot of annoying work. But these are all the different combinations we used. We tried promoters 1 through 7, 1 through 6, 6 and 7, just 6, 1 through 5 without an FNR box. Don't care, or sorry, you don't need to know what that is. And then just promoters 1 through 5. Long story short, as it turns out, just promoter 6 was the winner. So we took promoter 6, and we swapped out the natural promoter in front of this PDH complex. So now it should be anaerobic, and this would give us our 2 extra NADH. All right, so again, this was the winner because it gave us the highest levels of anaerobic growth. Okay, so 1.75 compared to all the other combinations, which only one of them gave us a almost equivalent. That's promoter 6 and 7 as a combo. It gave us the highest levels of ethanol, and it gave us the highest enzyme activity too. If you folks ever have to do enzyme assays, I'm super sorry. I had to do too many of them, but basically, if you want to see if an enzyme's working, you have to do an assay to figure out, how active is that particular enzyme under whatever conditions you're checking. Oh, and this was really cool. This is back when this was new, and now it's just like commonplace. We also did what's called quantitative PCR, where it's a special type of PCR. We're not familiar. PCR is a method of basically copying genes in vitro in a test tube. Quantitative PCR, what we did here was actually measure the activity of an enzyme by quantifying how much mRNA is being produced. Okay, the more active a gene is, the more active or the more mRNA you get. So, long story short, that red line, that was our homoethanol mutant with promoter 6. That was the winner because it made an obscene amount of mRNA for that operon I mentioned compared to all the others. So what am I saying here? Okay. What we did was we took E. coli, we deleted the genes for the competing fermentation pathways, so it should only produce ethanol. And then, to increase the ethanol output, we genetically modified the pyruvate dehydrogenase complex that handles that transition step to double the amount of NADH, so you should get lots of ethanol. And that's exactly what we did. So these are growth charts. They're showing you cell growth, and it's also showing how much ethanol is being produced, or at what yields. So we'll get to the good stuff here. So on the left-hand side, this is the wild type. That's the original organism. Okay, it was giving you a 16% ethanol yield from the sugars you were feeding it. Our strain SZ420, our homoethanol mutant, was giving us a 90% ethanol yield. So we know what we did was working. So that led to some further challenges that other grad students took because I went to a different direction. So yeah, this was great. We had this new mutant. It was making lots of ethanol, but there were some challenges. For one, I don't know if you've heard of what gram-positive versus gram-negative are. E. coli being gram-negative isn't a big fan of high concentrations of solutes. So we initially couldn't give it very much sugar because it would just die from osmosis. Okay, so one of the grad students, actually I think 2 or 3 of them carried the torch, did an artificial selection experiment where we had these small-scale bioreactors where we maintained continuous E. coli cultures. And every day or every other day, we would transfer it to a new bioreactor that had just a little bit more sugar in it. So over a period of 5 years, we went from about a 1 to 1.5% sugar solution to when I graduated, it was almost at 12%. Okay, 12% sugar is obscene. That would kill most other bacteria, but through artificial selection, we just gradually pushed it and pushed it and pushed it. So in the end, we were ending up with something shy of a beer. So it was like around 5-6% ethanol. It would be absolutely horrible beer, but still, it was there. Now, I mentioned how some folks like using yeast instead of bacteria. Yeast will produce lots of ethanol before they die from it. They can go as high as 15% or more. Right. Kids are in the garage banging on the wall. All right. We will speed through the second half here. I promise I'll be brief. But this first project actually went pretty seamlessly. We didn't run into very many hiccups, and it took about 2 years. It really, really wasn't bad. Where things got nasty is where I took it after that. Because what I wanted to do next was I wanted to take this homoethanol mutant. I want to say, okay, how can I genetically manipulate this organism to produce something other than ethanol? A different reduced fermentation product, another fermentation product that requires NADH, but it's not ethanol, something else valuable. And I decided to roll with sugar alcohols. Okay, xylitol is the most common one. It's in a lot of chewing gums. It's cheap. It was easy to work with at the time. But there are a lot of organisms out there that make very rare and expensive sugar alcohols. And I basically wanted to create a template organism where I could take those genes that make rare sugar alcohols through fermentation and pop it into my easy to work with E. coli. And then I also wanted to increase the NADH output to its maximum levels. And that's where I almost quit. It was really bad. All right, so here's the overall goal. Okay, so instead of making ethanol, what I'm going to do is I'm going to turn xylose, a 5-carbon sugar, into xylitol. And then, actually, I'm saying this wrong. So, all right, I'm flustered. I'm trying to get through this quick. So my metabolic goal here, we want to take E. coli, grow it on 2 different sugars at once. 6-carbon glucose, 5-carbon xylose. Okay. The reason being is that, again, when you break down biomass, you're left with a mixture of 5 and 6-carbon sugars. So here, the 6-carbon sugars would be broken down to produce 10 NADHs, called reducing power. And then those 10 NADHs would then go to the 5-carbon sugars to be converted into sugar alcohols. So ideally, 1 molecule of glucose would have enough reducing power in it to give us 10 molecules of a fermentation product, which is pretty impressive yields. All right, so bacteria don't really make sugar alcohols. That is actually something commonly done by yeast. I worked with DNA from a yeast called Candida boydini. So I actually extracted the gene responsible in it for making xylitol from xylose, and I transferred it to E. coli. How I did that was by inserting the gene into a plasmid and then putting that plasmid into E. coli. All right, now, because I wanted only making xylitol, I had to delete those genes for making ethanol, so I did do that. And I knew it was successful because those mutants that showed up missing that gene wouldn't grow anaerobically anymore. So I guess I made an obligately aerobic E. coli, which is kind of weird. Yeah, those are those teeny tiny tubes I was mentioning where we do the growth curves. Those things still haunt me. All right, next hurdle I had to deal with. Okay, I want glucose to produce NADH. I want the xylose to be turned into xylitol. Here's a problem with E. coli. If glucose is present, it won't touch any other sugar until it's done with the glucose, right? It's that kid that wants to eat like the good stuff first and not touch the vegetables. The xylose was the vegetables. Okay, so we had to make it more appetizing. So very, very long story short, we had to eliminate what's called catabolite repression. We had to tell E. coli that it's okay to eat both the sugars at once. So we did that by deleting the gene that's responsible for repressing the genes for catabolizing other sugars. And I did do that. And the neat thing is that what that chart's trying to show here is that now they were consuming both the sugars close to simultaneously instead of one at a time. All right, next thing I had to do, okay? E. coli can use xylose as a carbon source. I didn't want it to anymore. I wanted to turn the xylose into xylitol. So I deleted the genes responsible for breaking down xylose. Why I had to delete more than one, I still don't know. My advisor gave me bad advice. I deleted the wrong one first, and then when we went back and deleted the other one alone, it didn't work right. So I went with deleting both, and then it worked. Science. Now, what almost broke me. Engineering an anaerobic TCA cycle. Okay, so folks, that TCA cycle where acetyl-CoA enters and gets fully oxidized and get those NADHs, FADH2s, and ATPs, it is not fully functional under anaerobic conditions. And the problem is these three reactions at the bottom of the cycle. They're all tied into the same operon that, similarly to that PDH complex, actually scary similar, is regulated the same way. Oxygen's not around, those genes aren't being active. So I had to figure out how to turn that operon on to maximize anaerobic NADH output. So, if it ain't broke, don't fix it. If a promoter replacement worked before for that transition step, why wouldn't it work here? And that was, again, what almost broke me, because all I had to do was a promoter replacement using the same promoter from before. And folks, this is the dark side of grad school. I repeated the same experiment that had worked a million times before for somewhere around 6 to 7 months, every day, repeating the same thing, beating my head against the wall, worrying that I wouldn't graduate. I still remember when I finally got one mutant where the promoter replacement worked. I was running up and down the hall screaming like a fool because I had it. But yeah. So massive operon, especially for E. coli. I swapped out the promoter with that same promoter 6 from before, and sure enough, it worked. It worked great. So I'm the first person to engineer an anaerobic TCA cycle. And I did enzyme assays. All the genes in the operon, their activity was through the roof under anaerobic conditions. I did quantitative PCR. It was producing massive amounts of mRNA under anaerobic conditions. It was everything I could have hoped for. Yeah, here's the enzyme assays. There's my AI12. That was my final mutant. It was pretty ridiculous how high these numbers got for activity compared to the wild type. It was almost, quadruple for glucose, and what's that? 13-fold for alpha-ketoglutarate. And then I'd look at xylitol output, and that was where I kind of ended up. It was making some xylitol, wasn't making a ton. Someone else was supposed to take those reins from me, and I don't think they ever did. But where's that last chart? We'll use this one. So long story short, it was breaking down the glucose, giving me my 10 NADHs. And then those 10 NADHs should theoretically be going to xylose to make xylitol. And you may wonder, okay, did all 10 get used? No, my yield was somewhere around 6 to 6.5 NADH being used to make xylitol. Where the other 4.5 went, anybody's guess. There's other cellular processes that use it, but it is what it is. But yeah, this experiment was just showing that those outputs. So my xylitol to glucose yields, again, was it right about 6? Oh man, so further work. So this is actually based off of a presentation I gave to the USDA when I was applying there. So, I had to tell them, where can I go with this, even though I couldn't touch it because it belonged to the former school. But yeah, ultimately my goal was to create this template E. coli organism where, again, I could take fermentation genes from other organisms to make reduced products, pop it in there, and it's just going to crank them out. All right. Sorry about that. I haven't really gone through that in a long time.
I just have a quick question. So when you were spending those like 6 months, were you like trying to find the correct like promoter to get like how like were you just
So the method we used to replace promoters basically was the same that we used for knocking out genes. So instead of like knocking out the gene by physically removing it, we knocked out the promoter and swapped in the new promoter at the same time. We just kind of snuck it in as part of the process. It's a clever variation on it. I don't think many other people did. No, it was weird because I had deleted what, 5 genes. No, 7 genes. Deleted 7 genes using this method. We tried out all those promoters with the first half of the project, which was like 7 or 8 more combinations. I had done it successfully like 15, 16 times, but this one operon, and DNA is weird, right? Sometimes it wants to cooperate, sometimes it doesn't. That region just did not want to swap that promoter in. It was bizarre. I couldn't figure it out, but to confirm, you do a PCR react. You do a PCR. I always want to say PCR reaction, but my buddy Colin Jager yells at me every time I say reaction because that's what the R stands for, Andy. Every time you do a PCR, I would confirm doing a PCR. Okay, because I would amplify that promoter region, and what I would look for is one that was a different size, because the promoter region we worked with was bigger than the original. So I would run a PCR, I'd do gel electrophoresis, if you know what that is, and I'd look for my little bands of DNA. And like I said, I don't know how many times I did it. Like every day to every other day, I would run the gel and I would pray that to be one band that was a little bigger. And for months and months and months and months, I was just not getting that band. I was ready to give up. We were going to take it in a different direction. And finally, I got one that worked. I don't know why. I was playing the right music that day, or the stars were aligned, but one finally took, and that's all you need. But that is the essence of research, right? You may beat your head against the wall, getting something to cooperate, but when you get there, it's the best feeling in the world. As long as people aren't breathing down your neck about where's my grant money.
So it was just you trying to find that right promoter to like switch out.
It just didn't want to take it.
Is that what it was?
We
just didn't want to take it.
Yeah, the method wasn't working. The method we had done 16 times successfully before, just this one time refused.
So you had to try out different methods then?
No, it's just wash, rinse, repeat. It either worked or it didn't. We were kind of stuck there. And that's kind of the problem with metabolic engineering. It can be really unforgiving. Because if you can't... Go ahead.
Oh, so like then it finally worked. So then how do you make sure that that happens again? Because it wasn't working.
Yeah. I didn't have to. Once it's done, it's done. That's the beauty of it. Once you genetically manipulate your organism and it's doing what you want, you don't have to go back and mess with it. You go on to the next step. Yeah. Right. How do I put this? A lot of research is very investigative, right? I talk about a lot of my ecology friends. They go out in the summers, like my friend Allison Sacerdote. She's out in Lake County. She's studying smooth green snakes and blue-spotted salamander. She's out there right now. She's probably there right now looking at vernal pools. And she's collecting data all summer. And by the end of summer, she just does statistics to make sense of all that data. That's kind of traditional research, very investigative. We did, I don't know what to call it, Nelly, linear. I mean, it's just, there's a goal and you try to achieve that goal. And the problem is that it's unforgiving. Okay, with investigative, research, it could go all sorts of different directions. It just depends on how the data works out for you. It's not that way. That gene doesn't want to be deleted, you're screwed.
Yeah, I would say ecological research is very different than your genetic engineering research.
But even still, and the funny thing is, and I'll be candid here, when I sat down with my advisor that first day, he's like, okay, we can go one of two directions here. So I was his first grad student. He's like, we can do metabolic engineering, or we can get in on metagenomics. And metagenomics is basically trying to identify, say you have a soil sample. You can't culture the vast majority of the bacteria in that soil sample, but their DNA is there. And with metagenomics, you're trying to identify all of the organisms in an environment by looking at its DNA all at once. So that's, and sometimes I wonder where I would be if I went that direction, because right now, like human microbiome research is huge. And they're basically doing metagenomics, right? They take a poop sample and they screen what bacteria is in that person's fecal matter. But yeah, that would have been way more investigative than what we did. Yeah, you throw the term engineering with something, it's not going to be forgiving. That I didn't know at the time.
Yeah, it kind of it just seems incredible how, like, when you're investigating something and you can, like, you're kind of like seeing what I want to say the universe, what it results in. But what you're doing is just, I never thought there'd be so much trial and error. We're just trying to achieve this one specific goal, and the universe is just like, it's up to whatever happens. It's crazy.
Yeah, it's one of the things that scared me off of it, because the same week that they offered me a temp full-time position back in 2013, Nelly, the exact same week, I was actually offered a postdoc position with the USDA. And I had to make a hard decision that week. Which way am I going to take my future? But the thing that scared me off is that they wanted me to continue doing more metabolic engineering. But at the time, and I think I'm long past worrying about any sort of NDAs, they were doing work on engineering yeast to produce some precursor to an industrial adhesive. So basically cutting out some of the chemical synthesis, some of the cost there, by having the organism do half the work for you. And I was like, do I want to do this again? Also didn't help that guy had never had a postdoc before, and I just got done being someone's first grad student. So it's like, nope. And anyways, teaching's way more cathartic anyways, mostly.
Did they end up using, two questions. So the E. coli you engineered to produce xylitol, I didn't quite understand. So like I understand with ethanol, right, its use as a biofuel. What was the purpose of producing xylitol?
Xylitol was just cheap and easy to work with.
Okay.
Or really is just cheap. Like when I had to do HPLC, high pressure liquid chromatography, that's something I left out. We did a lot of HPLC work where, after the organism was grown, how did I know how much glucose was left? How did I know how much xylitol was in there? I used liquid chromatography, which told me how much was left in there. But yeah, mostly because it was cheap, and there were a number of yeasts that produce xylitol naturally. No, the idea was to create just what's the word? It's basically a blank template E. coli for producing any reduced fermentation product. Primarily, ideally, from 5-carbon sugars. So yeah, there's other rare sugar alcohols out there that cost way more money, that there are yeast out there that produce naturally. Just extract the genes from those yeast, pop it into a plasmid, throw it into my E. coli, and hopefully it would produce it. Yeah, that wasn't even the bad project. The bad one was the girl who did the butanol research. That was a nightmare. She had a another. She had a 6-month issue of her own, like I did. So, there's a group of bacteria called the Clostridia. You probably know botulism, tetanus, maybe gas gangrene. They make more fermentation products than any other bacteria, and their fermentation pathways are stupidly complex. They'll actually regulate making neutral products and then acidic products and then neutral products and acidic, and the local pH will actually control which rotation of fermentation products they're making. So they do not make an ideal organism for genetic engineering. Okay, E. coli is better. So she wanted to take their ability to make butanol and give it to E. coli. There's a lot of money in butanol. And that poor girl, I don't even know if she got to produce a tiny amount of butanol in the end, but it was a nightmare. She had to do PCR to copy this massive operon that Clostridia used to make butanol. It's like 8 genes. It's some obscene number. It's like 7,000 or 8,000 base pairs, which is big for bacteria, and that's huge for PCR. Like, people do PCR, they're copying like a 1,000 to 2,000 base pair gene. No big deal. You start getting to 8,000, things can get a little sketchy. She repeated that PCR for 6 months, failing. And the reactions were so long, she'd have to set them up at night so she wasn't taking up our thermocyclers all day long. And finally, I stepped in, and we actually got a paper out of it. We actually just started messing with the extension temperature, which we've had talked about PCR before. The Taq polymerase you use is supposed to work at a high holy 72 degrees. It's not necessarily the case, as I found out. You can lower it. The problem with Clostridial DNA is that it is very, very high in adenine and thymine and very, very low in guanine and cytosine. So as a result, it's very sensitive to breaking up when temperature goes up. But she managed to copy it. She gave it to E. coli, and it barely worked, but she was just a master's student at the time. That was a really bad project. The paper everybody seems to cite is the lactic acid work he did after I left. They switched to doing homolactic acid fermentation, and those papers have been cited a million times. I don't know why. But that was my advisor's big claim to fame. He engineered an E. coli, and this is for the chemistry folks. You talked about stereoisomers at all? You get D and L forms of organic molecules, right? Left-handed, right-handed versions. When you make lactic acid commercially, you get a mixture of both D and L forms. And the L form is way more valuable than the D form, but it's really expensive to separate. He engineered an E. coli strain that makes only the L form. And that was his big patent cash cow. Then his advisor, because there's always these pedigrees, his advisor was, can't remember his name anymore, but he would get hundreds of millions of dollars in grants to build these huge biorefineries. Guy was crazy. But anyways.
I have a quick question. How did you decide which biocatalyst you were going to use?
So we went with E. coli mostly because how easy it was to genetically manipulate, but 2, because it was cheap and just overall easier to work with. Yeast are great. Yeast will produce a lot of fermentation product, but when it comes to genetic manipulation, because it's a eukaryote, it's a little bit more difficult to deal with, and there's more paperwork involved when you start genetically manipulating eukaryotes and not prokaryotes like bacteria. They can also be pickier eaters than bacteria like E. coli. Yeah. Oh, and then there's more challenges too. And then, a lot of good fermentation products are made by other bacteria. I will tell you, it's much easier to convince one bacteria to produce products from a different bacteria than it is convincing a eukaryote to make a fermentation product from a prokaryote. you run into genetic issues like introns and exons, which bacteria don't have, but eukaryotes do. Oh, thank you for coming. Yeah, another direction we almost went is focusing on engineering E. coli to break down biomass, cellulosic biomass. So like feeding E. coli like raw corn leaves and saying, just raw corn kernels and or like elephant grass and say, okay, you break this down. But E. coli doesn't have the enzymes to do that. Who does is the Clostridia. Those same nasty bugs, they produce cellulases. They're actually really good at breaking down plant material. So at one point, we were tempted to start engineering E. coli to produce cellulases so it can do everything all at once, but it's a bit of a pipe dream. Normally, it's a two-step process. You have an unrelated organism, or you use enzymes to break down the plant material, and then the residue you get left over, that gets fed into the other organism that makes the product. And all-in-one is great, but realistically, I think it'll forever be kind of a two-tier approach.
Well, thank you so much.
Yeah, that project was great. When early on, things just blazed by. Like we knocked out those 4 competing fermentation pathways in like a couple of months. It was a joke. And then it seems like the more we tweaked the organism, the longer things took until I got to that last promoter replacement that took me 6 months. But yeah, that was great. I mean, we didn't have to go back and fix anything with that. It was just boom, boom, boom. We did HPLC analysis. We did the enzyme assays. The data was beautiful. Yeah. No. I know my advisor really wanted to, but right around the time I was leaving, 2 of the master's students switched to PhD projects or PhD status, and switched to PhD students. So they got their honorary masters and like, okay, keep going. And they just took their projects and they pushed it further. One of the students was working with one of my advisor's old lactic acid mutants, and he was trying to push that one further and get it to make more lactic acid. I cannot remember what Aaron did. Yeah. Otherwise, we always had master students working on those artificial selection experiments. Those were really easy. Just involved coming in and freshening up the media and just collecting more data. But yeah, I don't know. I don't even know if he has any grad students anymore. After Ryan and Aaron graduated after me, I know things slowed down in there a lot.
Well, thank you.
Welcome. Yeah, so I blazed through that. That's meant to be like an hour and 15 long presentation.
Well, you made it easy to understand, so
I tried.
you did good.
Thank you. I appreciate it.
Yeah.
Thank you, Andy.
Welcome, Nellie. Where are we at with our favors? I can't remember. I'm just kidding.
I think I owe you.
Okay.
I think I owe you like 10 at this point. So
No.
I'll have to bring you some more Nuclearis beer.
Okay, fair
You
deal.
guys didn't hear that, but
Yeah, this is our bartering system.
All right. Well, I'm gonna head out. Hope
On
everyone
that
has a good
note,
day.
later, folks.
Yeah.
right, next week we're on campus next week, right?
Yeah.
Okay, all right.
Gonna
Present.
present.
Okay.
Yeah.
All right. Well, I'll see you guys in person next week then.
That's good. All right. Thank you.